Hey everyone, Dr. Mungli here. So in this video, I will be explaining regulation of liver and muscle glycogen stores. I have a detailed video on glycogen metabolism regulation. The link for that video is appearing in the upper right corner right now, and also I have given link in the description below. So let me just quickly explain, and also to be very brief in explaining all the regulators, response of tissue, and the state in which this liver and muscle glycogen stores are regulated. So all that point I am going to cover in this particular video. So in order to explain that, so I have selected liver here and the muscle, skeletal muscle. So liver, in terms of quantity of glycogen per gram ratio, we have high concentration of liver glycogen in the liver. In terms of total quantity of glycogen in our body, so it is there in the skeletal muscle. So in order to understand how these glycogen stores are regulated in the liver and the muscle, we need to consider certain scenarios here. So the scenarios that I am considering, first here is the fasting condition. So under fasting condition, what will happen to the stores of glycogen in the liver? So as you all know, under fasting conditions, so we have increase in the hormone glucagon. So the hormone glucagon is increased. So increase in glucagon is a regulator of glycogen stores in the fasting condition. So when there is increase in glucagon, you know there is a decrease in insulin levels. So insulin levels are decreased. So basically under fasting condition, insulin is decreased and glucagon is increased. So now increase in glucagon. So glucagon will bind to glucagon receptor and it will increase cyclic AMP molecule. So cyclic AMP is the second messenger for glucagon, and there is increase in cyclic AMP in the tissues. So because of this, what will happen? So as you all know, cyclic AMP is going to activate protein kinase A, and that protein kinase A is going to activate glycogen phosphorylase kinase, and because of the activation of glycogen phosphorylase kinase, there will be activation of glycogen phosphorylase, and thereby glycogen degradation will increase. So there is increase in glycogen degradation. So that's what happens under fasting condition: increase in glycogen degradation and decrease in glycogen synthesis. Because under fasting condition, glucose is not available sufficiently, that's why. And also it's all about glucagon, so which is keeping your glycogen phosphorylase active, that's why glycogen degradation is increased, whereas insulin is down here and it's a fasting condition, that's why glycogen synthesis will be decreased, because the cyclic AMP protein kinase A is going to phosphorylate enzyme called glycogen synthase, and that glycogen synthase is inactive whenever it is phosphorylated, whereas cyclic AMP protein kinase A is going to phosphorylate glycogen phosphorylase enzyme, and the phosphorylated form of glycogen phosphorylase enzyme is active and it's going to degrade glycogen. That's what happens under fasting condition. Now let's move on to see what will happen after carbohydrate meal. So whenever we take a carbohydrate meal, you know there is blood glucose increases. That means there is increase in increased release of insulin from the beta cells of pancreas, and accordingly, so there is a decrease in glucagon hormone. So glucagon hormone is decreased, insulin hormone is increased, and also there is increase in blood glucose level. So blood glucose level is increased, and what will happen to the tissue cyclic AMP level? So because glucagon is decreased, so there will be a decrease in cyclic AMP. So cyclic AMP levels will be decreased. So this is what happens under fed condition, that is carbohydrate meal. So increase in insulin, increase in glucose, decrease in glucagon, decreasing in cyclic AMP. So whenever there is a decrease in cyclic AMP and increase in insulin, so what insulin does? Insulin is going to activate an enzyme called protein phosphatase, and this protein phosphatase, what it does is going to remove phosphates from all these enzymes. So when the phosphate is removed from the surface of glycogen synthase enzyme, so glycogen synthase enzyme becomes active, because glycogen synthase is active under dephosphorylated conditions. So when the glycogen synthase is active and glucose is sufficiently available, so there is increase in glycogen synthesis. So glycogen synthesis will be increased, and obviously there will be decrease in glycogen degradation. So this is what happens after carbohydrate meal. Now let us see what happens after exercise or during stressful situation. So whenever we are in doing exercise or when we are in stress, you know, there is increase in epinephrine. So hormone epinephrine levels will be increased because it is a stress hormone, and this epinephrine can bind to beta receptor on the liver and thereby increase in cyclic AMP. Second messenger cyclic AMP can be increased, and if epinephrine binds to alpha receptor, it will increase calcium, thereby it can bind with calcium calmodulin. So calcium calmodulin mechanism will increase here. So these are the two second messengers coming from epinephrine: increase in cyclic AMP, increase in calcium calmodulin. Overall, when there is increase in cyclic AMP or calcium calmodulin, so they are going to keep glycogen phosphorylase active, thereby they increase glycogen degradation. So there is an increase in glycogen degradation during stressful situation and during exercise, and when there is an increase in glycogen degradation, there will be decrease in glycogen synthesis, because cyclic AMP and calcium calmodulin they are going to have a negative effect on glycogen synthase and will have a positive effect on glycogen phosphorylase. This is what happens in the liver under three conditions here: fasting, carbohydrate meal, and exercise. Now let us move on to see what will happen in the skeletal muscle whenever we are in fasting condition. When we are in fasting condition, but note that when we are in fasting and restful situation, like fasting and resting, during that time, so fasting, so there is a decrease in insulin, so insulin levels will be decreased and glucagon levels will be increased, but we don't need to write glucagon here because muscles don't have glucagon receptor, so it's not relevant here. So there is decrease in insulin under fasting condition. When there is a decrease in insulin, so obviously in the skeletal muscle there will be decrease in glycogen synthesis. Glycogen synthesis is decreased, and also person is in restful condition, that's why, so glycogen degradation is also decreased. So there is decrease in glycogen degradation. So although person is in fasting condition, doesn't mean that in the skeletal muscle you break glycogen, because person is in resting condition, fasting but resting. So when the skeletal muscles are resting, that means you don't need to break glycogen. So both glycogen synthesis and glycogen degradation is decreased here. Now let us see what happens if a person takes carbohydrate meal but resting. So so blood insulin level will be increased here because person has taken carbohydrate meal. So there is increase in insulin from beta cells of pancreas, and what this insulin does. So as you all know, insulin is going to stimulate GLUT4 transporters, and GLUT4 transporters come up to the skeletal muscle, thereby it will increase glucose transport into the cell. So glucose transport into the skeletal muscle will increase because carbohydrate has person has taken carbohydrate meal, and insulin is increased, means blood glucose will also be increased, and this glucose will enter into skeletal muscle because GLUT4 transporters are brought up to the skeletal muscle membrane. So when there is increase in glucose getting into skeletal muscle, so obviously there is increase in glycogen synthesis. Glycogen synthesis will increase, and the opposite process, that is glycogen degradation, will decrease. Glycogen degradation will decrease. So they decrease in glycogen degradation. This is what happens. Now what will happen when the person does exercise? So skeletal muscles. So what happens during exercise? As we have already seen, so exercise increases epinephrine, hormone epinephrine is increased, and as I told you, epinephrine is going to increase cyclic AMP levels, and also when the person is exercising, so there will be release of calcium from the sarcoplasmic reticulum. Calcium will bind with calmodulin. So calcium calmodulin process, and also when there is increase in epinephrine, so there is increase in cyclic AMP, calcium calmodulin, and also ATP will break into ADP, ADP will break into AMP because exercise. So there is utilization of ATP there, that's why ATP to ADP, ADP to AMP. So there will be increase in AMP levels in the skeletal muscle. So, overall during exercise the regulators are, so increase in epinephrine, increase in EMP, increase in cyclic AMP and increase in calcium calmodulin. So, overall what happens because of this? So, the glycogen degradation increases, glycogen synthesis decreases. So, decrease in glycogen synthesis, increase in glycogen glycogen degradation is increased, and also there is increase in glycolysis. So, the breakdown of glucose into pyruvate and into acetyl CoA, that will increase because you know AMP levels are elevated during exercise, and AMP is a positive modulator on rate limiting and regulated enzyme in glycolysis called PFK1, phosphofructokinase 1. That is why glycolysis is increased. So, this is how this is how the liver and glycogen stores are regulated by variety of regulators under different conditions. So, let me quickly revise this here. So, here is your state, regulator and tissue response. So, we are taking example of liver and skeletal muscle, and liver we are looking at three conditions. One is the fasting state, carbohydrate meal, exercise and stress. So, whenever we are in fasting condition, what happens in the liver? Fasting means increase in glucagon, decrease in insulin, and glucagon bind to glucagon receptor, adenylate cyclase is activated, it increases cyclic AMP will activate protein kinase will activate glycogen phosphorylase kinase, and that will further activate glycogen phosphorylase by phosphorylating it, and glycogen phosphorylase, if it is active, it is going to degrade glycogen. So, there is increase in glycogen degradation, and glycogen synthase will be inactive because of phosphorylation. So, there is decrease in glycogen synthesis. When we take carbohydrate meal, what happens in the liver? So, insulin levels will be increased in the blood, regulator is in insulin, glucagon is decreased and blood glucose levels are increased, and when glucagon is decreased, cyclic AMP levels decreases. So, what insulin does? Increased insulin is going to activate protein phosphatase enzyme, and this protein phosphatase is going to remove phosphates from the enzymes. That means glycogen synthase is dephosphorylated and it becomes active. Glycogen phosphorylase, when it is dephosphorylated, it becomes inactive. So, glycogen degradation decreases, glycogen synthesis increases. When the person is exercising or in stressful situation, the regulator is epinephrine. So, epinephrine levels increased, epinephrine binding to the beta receptor on the liver will increase cyclic AMP again through adenylate cyclase and all that mechanism. And if the epinephrine binds to the alpha receptor, so it is going to activate phospholipase C enzyme that will break down PIP2 into IP3 and DAG, and IP3 release calcium from the endoplasmic reticulum. Calcium will bind with the calmodulin, and calcium calmodulin and cyclic AMP through protein kinase. It's going to keep glycogen phosphorylase active, thereby increases glycogen degradation, and it keeps glycogen synthase inactive, thereby glycogen synthesis decreased. Coming to the muscle under fasting condition but restful situation, what will happen? So, there is a decrease in insulin because person is fasting. Glucagon is not mentioned here because it is not relevant here because muscle does not have glucagon receptor. Insulin decrease means there is a decrease in glycogen synthesis, decrease in glycogen degradation. Why glycogen degradation is decreased even though person is fasting? Because in the muscle, when the muscles are resting, you do not need to break glycogen because there is no need of energy there. That is why glycogen degradation is not really increased. Carbohydrate meal but person is resting. So, what will happen? There is increase in insulin, increase in glucose. Insulin, what it does, it is going to increase glucose transport across the skeletal muscle membrane because it is going to bring up GLUT4, which is an insulin sensitive transporter. When glucose enters into the skeletal muscle, so glycogen synthesis is increased because glycogen synthase will be kept active by insulin. Glycogen degradation will be decreased. Now, when the person exercises at that time, so the hormone, that is epinephrine, is increased, and also this hormone, what it does, it is going to increase cyclic AMP levels, and also contraction of the skeletal muscle increases calcium release from sarcoplasmic reticulum, which will bind with calmodulin, and calcium calmodulin levels increase, So both cyclic AMP and calcium calmodulin mediated mechanism will overall keep glycogen phosphorylase active and glycogen synthase inactive, and thereby glycogen degradation is increased, glycogen synthesis is decreased, and also you can see glycolysis is increased because the AMP levels also will rise when we are exercising, because ATP is broken down into ADP, ADP into AMP, and AMP is a positive modulator on PFK1. Not only that, AMP is also sensitizes and acts positively on glycogen degradation enzyme, that is glycogen phosphorylase. So this is all about regulators of glycogen stores in the liver and the skeletal muscle. So as I told you already, like I have a detailed regulation mechanism in my previous video. The link for that video is appearing in the upper right corner. It is there in the description below. So please watch that. This video is just a brief review of my previous video in the tabular form, just to bring everything together, and I hope this video has helped you in understanding regulators of liver and muscle glycogen stores. So if you haven't subscribed to my channel, so kindly consider subscribing to this channel so that you get regular notification and updates as and when I upload a new video, and also make sure to click bell icon down there so that you receive a notification upon a new video being uploaded. So thanks again, and I will see you in my next video.